Dear students, welcome back, and we are going to discuss about the topics that are listed in Unit 2. And in this topic, we are going to discuss about structure of biomolecules. What are biomolecules? Remember four: carbohydrate, protein, lipid, and nucleic acid. So we are going to discuss about the structure of biomolecules, that is carbohydrate, protein, lipid, and nucleic acid. Nucleic acid comprises of DNA and RNA. And followed by that, we are going to discuss about the metabolism of these biomolecules. And followed by that, the third topic is going to be photosynthesis. So we are going to divide this topic into three. One is structure, second is metabolism, and third is photosynthesis. Now, to start with, let us understand a very basic concept which we call it as biochemistry. So most of you should know about chemistry. Chemistry is a process in which the molecules will react with one another. Whereas biochemistry, the same chemistry occurs inside a living system. We call it as biochemistry. Living things requires millions of chemical reactions. For what? To survive. And these chemical reactions will happen inside our system, and we call that as metabolism. Metabolism are of two types. One we call it as anabolism, another one is catabolism. I'll be discussing in detail about what is anabolism and catabolism, but together anabolism and catabolism is called as metabolism. And we usually have organic materials or molecules in a living system. We had discussed that in Unit 1. If you can remember that, please go back to Unit 1 video so that you can able to understand about what is this organic molecule that I am referring to. It is always made up of carbon, and these molecules are going to be usually large, but in certain cases it is also seen as a small molecules in a living system, and all these molecules will have a covalent bond. That means a sharing of electron will be seen. So we call this as organic molecules. Now we'll start to discuss about first a complex molecule called as polymers. Why should we call it as polymers? Poly refers to many. Many means large. So the large molecules are always referred as polymers, and these large molecules has to be synthesized from small molecule, isn't it? So we call it as monomers. So that means polymers are built from smaller molecules called as monomers. And we can, biologists, we also refer this polymer as macro. Macro here refers to a big, large molecules. So polymers, macromolecules are one another the same. What are the molecules, macromolecules that are seen in an organism's four category? Remember about biomolecules. One is carbohydrate, second lipid, third protein, fourth nucleic acid. Now let us look one by one. Before that, I have a table which summarizes the important key aspect of all these four macromolecules. To start with carbohydrate, we have a function of carbohydrate. As I told you, energy is the predominant primary source for carbohydrate because breaking of glucose, a process called as glycolysis, if you can remember, we had discussed that in the unit 1, leads to or gives us an energy source called as ATP. And this is how a structure of a carbohydrate looks. And this is an example for a monosaccharide. And examples of monosaccharides includes glucose, fructose. Okay, again for disaccharide means two simple sugars are joined together. So lactose and sucrose is an example. Polysaccharide means many or multiple. Example starch present in potato and cellulose present in plants. So this is an example for a macromolecules of a carbohydrate. Likewise, we have lipid, protein and nucleic acid. The simple form of lipid is going to be a fatty acid. Okay, along with that, glycerol. Where can we see the lipids? It's rich in oil and ghee, a good example for you to understand. And why this lipid is used for? Long-term storage of energy. Not only that, from lipid only we get hormones, especially steroid hormones. As you know, testosterone in male and estrogen in female comes from fats. And for protein, simple form is going to be amino acid, and the structure of an amino acid is shown here. Okay, and with the help of protein, we have lot of other classifications which is referred as enzyme. So lactase is a enzyme which is abundantly used by growing children, especially the children within the age of 1 will start to secrete lactase because they are dependent on mother's milk, right? So milk contains lactate. So this lactate is going to be broken down by the lactase so that the children's below the age of 1 will have good secretion of lactase. Again, lactase is an example for a protein. It's an enzyme, right? And followed by nucleic acid. We all know that it stores the information, and we call it as nucleotide, phosphate, and a base along with sugar is called as nucleotide. And an example for that is DNA and RNA. Now let us understand what is carbohydrate. Okay, so these are abundant molecule, organic molecule in nature. And what is the biological importance? That means what is the function? First important function is store of energy. I already told you, if you want an energy, you need glucose. And that is the reason why when you're so tired after a strenuous physical activity, you drink an energy drink, and the energy drink contains glucose. Apart from that, it also going to act as a structural compound for a cell membrane. Remember, when we discussed about a membrane of a cell, we know that it is made up of double layer, lipid bilayer, right? But apart from that, it also has certain protein onto which the glucose molecule is attached, and we call it as glycoprotein and glycolipid. Okay, so glycoprotein is also a part of glucose attached with protein. Like that, glycolipid means a lipid molecule attached with glucose. Okay, and what is the function of this glycoprotein? Involved in cell cell interaction. That means one cell to another cell, if it has to communicate, it will communicate only through glycoprotein and glycolipid, right? And the last important function is going to be the pyruvate, from which the degradation of glucose leads to production of pyruvate, and from pyruvate, your system can synthesize amino acid and lipid. Okay, there is a separate metabolism is there. We'll discuss that in a detailed way at a later point. Right. So, what is the chemical nature of carbohydrate? So, when I say carbohydrate, it means one carbon group and hydrate. So, it is made up of polyhydroxy alcohol along with the functional group, and we call that as aldehyde or keto group. Most sugars have a chemical formula. We call it as C, that is carbon, H, that is hydrogen, and O, that is oxygen. So we usually used to call it as C6, that means 6 carbon, H12, that is 12 hydrogen, and O6, C6H12O6, that means 6 carbon, 12 hydrogen, and 6 oxygen. And some of this carbohydrate may also contain the nitrogen, phosphate, or sulfurate. Okay, now how can we classify the carbohydrate based on how many units that it has? If it has only one unit, we call that as monosaccharide. Okay, we call that as simple sugar. Okay, but this monosaccharide examples are multiple, and followed by that disaccharide, that means two monosaccharide which is covalently linked with one another. The third one is oligosaccharide, a few monosaccharide covalently linked, which is usually 3 to 10 monomers. And the last one is called as polysaccharides, that is polymers consisting of multiple chains of monosaccharide or disaccharide. Usually more than 10 single unit monomers is considered to be polysaccharide. disaccharide. So this is the structure of a monosaccharide, and the general formula is CN H2O and N. Okay, so that means how many hydrogen, oxygen, and carbon will vary from one carbohydrate to another carbohydrate. It is also called as simple sugar. And we also have something called as isomers. As you can see here, all these molecules are of carbohydrate, but it will have same 6 carbon, 12 hydrogen, and 6 oxygen. But in spite of having same C6H12O6, the structure of hydroxyl group present in one molecule and the other molecule is going to be slightly different. As you can see here, we have an H here on the left hand side, and on the right hand side I have OH here. But whereas here on galactose you have OH on the left hand side and H on the right hand side. Okay, so there is a difference in the position on which hydroxyl and hydrogen ions join with carbon is varying. So we call this as isomers. Iso means same. That means formula wise same, but structural wise it is little different. So we call it as isomers. And we also have something called as epimers. As you can see here, we have D-glucose here, and this when you look at the structure, the OH group here is present on the right hand side on this structure, D-glucose, whereas on this glucose you can see that it is present on the left hand side. So there is the one specific carbon atom which is going to be different from one another, and we call it as apmers. So you can compare it with the D-glucose and D-galactose. And we also have dextrorotatory and levorotatory forms of monosaccharide. So when you pass a polarized light onto the solution of this carbohydrate, this structure, as you can see here, D-glucose and L-glucose, this will start to rotate. If it start to rotate on the left hand side when the polarized light is passed, we call it as levorotatory. That means you can see L-glucose. You see that OH is on towards my left hand side. And when you pass the same polarized light onto the glucose, in case if it is a D-glucose, it will start to rotate on right hand side, and that is the right reason why you have OH on the right hand side. So we call it as D-glucose. Now you should be able to understand what is called as levo and dextro rotatory, because on presence of polarized light, this will start to rotate. And what is disaccharide? Again, it's a source of energy, but this disaccharide has to be converted into monosaccharide, and then it can be converted into a energy source directly. So these are of two monosaccharides which are joined together by a glycosidic bond, which is very important. Okay, so this glycosidic bond will join these two monosaccharides together with one another. And examples are shown here: maltose, lactose, sucrose, cellulobiose. All are going to be the example. Okay, so on maltose you have glucose and glucose, and the bond that is formed is going to be alpha bond, which is formed between first carbon to fourth carbon. So we call it as glycosidic bond. So maltose consists of glucose and another glucose. So where it is present? It is present in germinating seeds, especially barley. And lactose, this is composed of galactose and glucose, and the bond that is formed between this glucose and galactose is going to be beta 1 carbon and 4th carbon, and still we call it as glycosidic bond. And where it is present? Milk. Lactose means remember the milk. Like that we have sucrose and cellulobiose. The last one is called as polysaccharide. These are monomers but are joined together. As I told you, more than 10 monomers joined together is called as polymers. Okay, it is long in length. Okay, so we call it as polymers. So these monosaccharides are joined together by a glycosidic bond. This polysaccharide can be further classified into homopolysaccharide and heteropolysaccharide. As you can see here, a pictorial representation is shown here. Homopolysaccharide means all the saccharides are going to be of same type, whereas heteropolysaccharide, the carbohydrates that is going to be seen here joined with one another is going to be of multiple different types. So we call it as homopolysaccharide and heteropolysaccharide. So when you hydrolyze, when you break the homopolysaccharide, you will get the same kind of monosaccharide. Why? Because all are made up of same monosaccharide. Whereas if you break the heteropolysaccharide, you will get more than one type of monosaccharide because all these are going to be different monosaccharides. Okay. Example for homopolysaccharide is starch. What it contains? It contains amylose and amylopectin. And a good example for source of starch is going to be potato, right? And apart from that, we can also give a glycogen as an example, which is present in animal. Starch is present in plant. Okay, both are homopolysaccharide, and glycogen is made up of glucose. Okay, multiple glucose joined with one another, and it is joined in alpha 1,6 bond. Okay, so we have a bond that forms during the formation of glycogen. And another example is chitin. A good example is these are present on the exoskeleton of arthropods. Arthropods means prawn, crab. Those will have this chitin, and it is a polysaccharide, homopolysaccharide, which consists of N-acetyl glucosamine, which is be joined together by 1 to 4 beta bond. So we call it as glycosidic bond. So chitin is also an example for a homopolysaccharide. And heteropolysaccharides are different types of monosaccharides. The good examples are shown here. Heparin, most of you should know, which is a blood coagulating agent. What it is made up of? Glucosamine and glucuronic acids. Okay, which is function is to prevent the clotting of blood. Okay, so and where these heparins will be present, it is present in the mast cell. And another good example is agar, okay, which is composed of galactose and hydrogalactose, and which is a structural component in algae. Okay, and it is present on the cell wall of the algae. Apart from that, we have pectin and hyaluronic acid. I hope this will make you understand the complete details about carbohydrate. In the next topic, we'll start to discuss about the other biomolecules like protein, lipids, and nucleic acid in detail. Thank you.